Hello, everybody. Welcome to another episode of Redefining Society podcast. I am very excited today to have with me Dr. Neal Baer. And we were talking before the show, and I was talking about my other podcast, Audio Signal, where we talk about storytelling, because we're here to talk about a book that he wrote, and it's about health and technology and the impact that it has on society. And I think we're going to put our philosophical hats on quite a bit during this conversation. But we were also talking about how Neal here is origin story. Let's say it is about media, and he has participated in writing and producing a TV show connected with the healthcare world, and I'm sure you heard some of them. So we'll see. Maybe we'll have another episode after this, but let's focus on this one. I would like for you to introduce yourself to our audience, and then we'll talk about your book. I'm very
Great.
curious.
Thank you. Well, it's really nice to be chatting with you from New York City. I live in Paris and in Boston. And that's because in Paris, I work now as a TV writer, producer, what's called a showrunner, overseeing some shows, one in Poland, one in Norway. But I did the shows ER, Law and Order Special Victims Unit, for a total of 18 years, the first 7 years of ER, the early years of SVU, 11 years, and then other shows, Under the Dome and Designated Survivor. And I created the first master's degree program in media, medicine, and health at Harvard Medical School, where I teach master degree students how to tell stories to have a forceful impact on improving health, be it equity, access, well-being. And so we use storytelling to do that. So through any kind of art modality you can think of, from poetry and sculpture to dance and documentary. So my book, The Promise and Peril of CRISPR, comes out of my work as a TV writer, but also as a physician. I'm a pediatrician, and I attended Harvard Medical School. And then while I was on ER and SVU, I was doing my internship in pediatrics at Children's Hospital Los Angeles. So I was very lucky to, explore these different realms for my career, and I brought it all together for the book that I'm doing that was just published by Johns Hopkins, and also for the master's program that I do. So I was really interested in CRISPR because as a pediatrician, I saw very, very terrible diseases like sickle cell disease, adrenoleukodystrophy, Tay-Sachs, that affected children and often caused profound pain, certainly with sickle cell disease, and certain death with Tay-Sachs and adrenoleukodystrophy at a young age. And so I was motivated to look at all the treatments that were down the road, in the future, and what we could do now. I'll never forget the night I was on call at Children's Hospital Los Angeles, and I had a patient with sickle cell disease who had been transfused wrongly, unfortunately, because it was buried in an old paper file when we still had paper, medical records, not to transfuse her. And she had been transfused, and so we were, madly trying to save her. Unfortunately, we did, with the brilliant help of our attending who came in the middle of the night. But these children suffer horribly, horrible pain, strokes often, shortened lifespan. So it's a really terrible life. And lo and behold, we were becoming closer and closer to finding a way to treat them and cure them. And that was through CRISPR. And now we can. And so we can cure people with sickle cell disease by editing their genes.
And you're getting there because it's an acronym, and I work a lot in cybersecurity, so there is a lot of acronyms, and sometimes people hear them in the news and they don't know exactly what it is. So I did a little research, and I know that it stands for cluster regularly interspaced short palindromic repeats, which honestly, to somebody like me, doesn't say much. So in a few words, you had to explain what it is and what it does. And you started talking about the application. So, kind of like a 101 on
Right.
CRISPR.
Yeah. So CRISPR, really think about when you had a word processor before it automatically fixed your words. Think about how if you typed a word incorrectly, like house, Huose. Think about how in DNA we have, 4 letters that code for proteins and everything that is made by our DNA, so that in certain genetic diseases, there may be just a few letters that are off, or even one, and that can cause alarmingly awful diseases. So with CRISPR, we have a technique where we can home in on the incorrect DNA, those letters, use enzymes called Cas9. Now there's many other Cases to cut that precisely, and they're getting better and better at it, because in the past there have been off-target effects, which we can talk about. So it goes in, locates that piece, cuts it, and then glues in. So it's a glue, cut and paste, correct letters, correct genes for the mutation that's there. It's easier to do for blood-borne diseases like sickle cell disease and beta thalassemia, because you can deliver it to the blood. Much harder to figure out how to do this for cystic fibrosis or Duchenne's muscular dystrophy, but they're working on it. And so beta thalassemia, sickle cell disease, some rare protein folding diseases of the liver, because blood is, the liver cleanses the blood goes into the liver. These have been done through CRISPR. So essentially what CRISPR does, like for something like sickle cell disease, is when we're in utero as a fetus, we breathe through our blood, essentially, because our lungs aren't working. We're in amniotic fluid. So we have fetal hemoglobin, which really attaches to the oxygen that we get from our mother's blood. And then when we're born, that fetal hemoglobin turns off, and what then is on is our normal hemoglobin. Well, the quote normal hemoglobin for someone with sickle cell disease is not normal, because they have the mutation, those few letters. So we have figured out how to turn on that fetal hemoglobin again, and that kind of floods the body with the good hemoglobin, and these people are doing well. Now they have to go through a process of ablation and things like that, but essentially they get their fetal hemoglobin turned back on by CRISPR, by cutting and pasting in the gene that will turn it back on. Well, there are 100,000 people in the United States, Black Americans mostly, who have sickle cell disease. Shortened lifespan, life of horrible pain, strokes in children, because the blood is sickles and it gets caught in joints and in arteries. So this is amazing, because there are so many people who could benefit. It costs between $2 and $3 million to treat one person. It costs $1.6 million in someone's lifetime with sickle cell disease to be treated, often through Medicare, Medicaid, and $50,000. And I'm taking all these data from recently published studies. $50,000 out of pocket for many people who can't afford it. So this raises a fundamental philosophical, ethical question of who gets CRISPR. And so that's going to come down more and more as more of these these genetic engineering products are made. So we do in our book, The Promise and Peril of CRISPR, talk a lot about who gets this miraculous treatment and who doesn't. And we're at the beginnings, and I think it's important that we're starting to talk about this now as opposed to in the future when it's already happened. We have a wonderful essay by Ben Hurlbut, who is a professor at Arizona State University, who talks about how often science leads the ethical discussion. So what do I mean by that? Well, in 2015, when there was a lot of discussion first starting about CRISPR, when it had been developed by Jennifer Doudna and Emmanuel Charpentier of France, who won the Nobel Prize for it, there was agreement that we shouldn't do a certain kind of editing, which means we shouldn't edit embryos, which could then pass on the changes to progeny, to their offspring. And so I want to also make clear that there are two kinds of CRISPR. There's somatic and germline. Somatic is what people with sickle cell disease would receive. It is cutting their gene that makes the fetal hemoglobin turn back on. But if they had children, they're not passing on the, quote, non-sickle cell or good gene. They still would pass on the sickle cell trait because they have two copies. That's why they have sickle cell. They got one from their father, one from their mother. And they would definitely pass on one because they have both. And if their partner had sickle cell disease, there would be a much higher chance of them having sickle cell. It would be 50% chance instead of a 25% chance, because the mother, let's say, has two and the father has one, that would be half. So that's somatic. Germline is when we take an embryo before it's developed into a fetus and then a live birth, that if we go that direction, that then that person who's born has had their DNA changed before they're born. And that raises a lot of questions, as you can imagine, about the future, about enhancement and who gets this. And should we start CRISPRing embryos so that we don't have certain genetic diseases anymore? And where do we draw the line between a genetic disease and something like Down syndrome or deafness? And who's going to make those decisions? Right. So that's the difference. One can be inherited, one is not. And almost everyone I know is in favor of the somatic,
right?
Changing lives that are really tough and suffering versus changing human development or maybe human evolution. And so that's really where that's what the book is about. Like how far.
And that's the big question where I said we're probably going to get philosophical here, because there is no way. I had way too many of this conversation about technology and society and put my, sociologist hat on and think philosophy and think every new technology. Let's look at AI. Everybody talks about AI, generative AI. There is the fears, going to take jobs. There is the singularity. I just finished reading Ray Kurzweil, The Singularity is Nearer, the new version of his book. And I wanted to bring it up because he talks a lot about, this transhumanism where you optimize the human body. And my question for you was actually going into how different or how connected is the CRISPR with DNA manipulation, and right. and that's, you went there. So I would like to go a little bit deeper into that, and what are your thoughts on that? Right? Where do we draw the line? And And when do we say, let's embrace technology and see where we go?
Right. Well, let's start with what I said earlier, which is 2015, we talked about societal agreement, consensus. And then Dr. He in China did his experiment on the embryo, which became twins, split into twins. And that's then the academies like of engineering, science, and medicine said, well, maybe there are times to do germline editing after Dr. He did it. And that's what Ben Hurlbut writes about. Maybe there are special circumstances. So I can tell you the major special circumstance is if a couple they're both deaf with the same genetic mutation, they're not going to have children who can hear. They're going to have deaf children. And so of certain kinds of deafness, not all kinds. So the argument is that if they want to have a hearing child, who are we to say they shouldn't? So we have an essay by Carol Padden from the University of California, San Diego, who's deaf, who said, I don't see deafness, I'm deaf, and I don't see deafness as pathological. I see it as human variation. And we're hearing a lot about human variation. Why? Because we're telling a story right now where we see Gus Walz, Tim Walz, vice presidential candidate, his son, who has some neurodiverse elements. And that's opening our eyes to diversity. So Carol says, who's going to make a decision about deafness? Now, Ben Hurlbut argues that once you go down this path, it's hard to go back. And then what's next? And what's next? And what's next? And so how do you think about, something like Down syndrome or different, what diseases like adrenal leukodystrophy or Tay-Sachs, which is always fatal, versus Down syndrome? Who's going to decide where you draw the line? Denmark provides prenatal care and amnio to every woman who's pregnant, and they have a very, very low rate of Down syndrome. So, a lot of people feel like we should eliminate Down syndrome. Should we do that or should we not? And the question is, who's to decide who's valuable as a human being or not? Yeah. And so that's a big question for us to consider. Also, when we start to talk about the promise of CRISPR and say sickle cell disease, which I embrace CRISPR for that, we have to be very careful because we don't know the future. We embraced fossil fuels and it changed society, it changed our culture, it changed policy, politics, wars. We embraced plastics and they are ruining the ocean, like something like 20 to 40 billion, tons a year. I mean, just incredible. So, the promise of plastics, the promise of fossil fuel, the promise of nuclear energy. We've seen the Peril, Chernobyl, 3 Mile Island, clogged oceans, dying sea life, climate change. How do we take what we know now about those changes and those, what happened before we jump into CRISPR and AI? And the problem is we don't know. We can't know. It's the future. So how can we be more careful? How can we be more thoughtful about how we approach these things? That gets into how do we control who gets the pieces to make DNA? There are companies that sell this. There's really not oversight. There's no international oversight. There's a scientist who made horsepox, which is related to smallpox. You don't need CRISPR to do this. Because I've been criticized, well, you don't need CRISPR to do this. Well, I know that. But CRISPR makes things easier, makes bioweapons easier to make. You don't need CRISPR to decide whether to implant an embryo that has Marfan's or sickle cell or whatever. You can go through IVF and eliminate the ones that have the genetic malfunction or the mutation, and they only implant eggs that don't. But that costs a lot of money, and most people can't afford it. So George Church from Harvard, who's a renowned scientist, says maybe we should do editing instead of somatic editing to treat the problem, like sickle cell. We should use germline editing to get rid of these genetic diseases. Now, why do we have genetic diseases? Well, it is evolutionary in many, many, many cases. So for instance, with sickle cell disease, if you have sickle cell trait, 1 out of 13 Black Americans do. Why is that? Well, evolutionarily, probably happened about 80,000 years ago. People with the trait are more resistant to malaria. So where Africa has endemic malaria, people started to evolve from the pressure of the malaria. Unfortunately, if you got 2 of those genes, then you died. But if you had 1, which more people have, 1 out of 13 Black Americans, that's a lot. Only 100,000, I say, that's still a lot, have it, 20 million in the world. Then those people are unlucky, but it still helps most people who are exposed to malaria do better. It doesn't prevent malaria. So I think I've read that with Dr. He in China manipulated and mutated the receptor for HIV in this egg that then became twins, because the father in China had HIV, and it's not really clear exactly why he did it. We know more than we used to about it, but there's a lot of stigma about HIV in China. But we also know that it's not very hard, it's not difficult to have a couple have a child where one of the parents or even both have HIV, because we can take the sperm, for instance, and we can make sure that we clean it, so that it doesn't have it's washed, so it doesn't have the HIV, virus. So you don't need CRISPR for it. And there's some evidence that if you mutate that particular receptor, and people who were exposed to HIV in the past who didn't get it were, in a sense, very lucky because they had that mutation. But it may make them more susceptible to things like West Nile virus, we're finding out. So evolution has a process that is affected by the world pressure. And so the pressure of malaria made, we think, sickle cell disease. So, when we're starting to tinker and play with genes and turn things off and on, like, should we should we use CRISPR to make people, not have, high cholesterol? Well, we know that there are people who have high family cholesterol. Maybe we should take out that gene, but how is that connected to other things? And how is that connected to the evolution of lipids? So we just don't know a lot about that, but we go in at this point and start to cut. So, we have to be careful. And in terms of enhancement, we can't make people stronger, smarter. That's an agglomeration of genes. We can't even make somebody's eye color. It's not one gene, like a lot of these genetic diseases, which is why CRISPR is so amazing for that. But there are things we can do, like we know kids who don't feel pain, who have a genetic mutation. Supposedly, Russia and China are doing research to make soldiers not feel pain.
Of course, Captain America.
So that's an enhancement. So that's the peril. So as we learn more about what genes control, we could presumably in the future possibly manipulate them and change human evolution. Wouldn't it be great if we could eat grass, like cows, and digest it because of, problems, with food sources and food insecurity.
So while evolution takes millions of years to do that one change, and Darwin, we know somebody survived, but two sisters, one develop in one way or another, and you carry the things that work. But as you mentioned, something that may work in Africa during a certain period where malaria is very active, which unfortunately still is, but at the time, but there's always the other side of the coin, right? And we just don't know. So I think the big question here when it comes to technology, advanced technology, nanotechnology, AI, and all of that, whatever you can think, is what you said. It's a balance, and who makes the decision? Because I think that's the key. Like, who makes the decision for others? I mean, I don't want to get political now, but normally there is some people that want to make decisions for everybody else.
Well, have you ever heard a politician talk about CRISPR?
No, exactly.
It's too complicated and it's too whatever, so they say. It is discussed. Believe me, it's discussed at DARPA. It's discussed, and there are provisions in laws that say you can't use CRISPR to manipulate embryos like Dr. Hu did. But is that going to really stop somebody? So
do you see a role in public discourse, public awareness? I mean, part of your job is to be a science educator and teach to other scientists how to do that.
Right.
Do you see the value in the democratic decision in public?
Yes, I think absolutely. And the challenge is, it's one thing for the academies to say we need societal consensus, and then we start to dig into what does that mean? Like, who's
51%?
Like, how many, or who's going to sit at the table? Who's going to go to the conference where they have these discussions?
Right. Yeah.
Is it going to be philosophers? Is it going to be somebody who is, laying pipes? Is it plumbers? Who? It could affect everyone. So how do we think about that? And that's really the challenge of CRISPR and all technology. How do we, we're in a terrible place where people question vaccinations. So we're seeing more whooping cough and we're seeing more measles. And we're pretty good at knowing a lot about measles and whooping cough. We're less good about knowing about COVID because it hasn't been around. It's novel. And so people in general want definitive answers from science. And I think one of the problems we faced with COVID is it wasn't definitive from the beginning. How could it be? It was novel. And so we were learning, and we're still learning about long COVID, and we're still learning about, the variations of the virus mutations and, what kinds of mRNA vaccinations work best, et cetera. So it's an ongoing process of sciences. And so we have to help people understand that, first of all, that it's not like we have the answer, and we can't actually say we have the answer because that makes people really uncomfortable when somebody dies from COVID or they have a vaccination reaction, which can happen. So we're in a place, compounded by social media where there's lots of mis and disinformation about COVID, for instance. And yet, if you talk about measles, we know pretty much the side effects, and we know what measles can do. We know what measles can do to a child, cause encephalitis, all kinds of things. So one is we have to really think about how we teach science, how we talk about science, that science is not just about, it doesn't start when you're, an adult and you can make your own decisions about your healthcare, or as a teenager, you can make it with your parents and your parents make it for you as a child. We have to start teaching about the process of science, the experimentation that's involved in science, what science means, what a hypothesis means in theory and all of that, and experimenting. We need to teach, and we need to use AI to help people understand it better. There are new ways to look at peer-reviewed articles through Cite and Scholarly and other platforms that help us really see an article in relation to other articles and see its impact, which is amazing.
Yep.
I wish I had it when I was like in medical school.
I know. I wish I had it when I was writing my thesis.
Yeah, I mean, I had all these like little books on endocrinology and pediatrics and
Yeah.
and now it's like your cell phone.
Yep.
Look it
Pretty
up.
much.
So it's like that's really changed things.
Yep.
But still, we need to really think hard about where we're going with things. We just can't say, well, science has advanced here with CRISPR, so let's just keep going, especially with germline editing. So that's what I worry about. I've been accused of, saying, well, there are other ways of doing sort of the same things that CRISPR does. That's true. And we should be talking about those things. And and so we bring up these issues that are not open to the public, really. So how do we do that, as you asked? So one way would be, and this is my program at Harvard, is through the arts and humanities. How do we talk about it? So in my show, Designated Survivor, the last season, season 3 on Netflix, deals with CRISPR and how it was being thought to be used to do some nefarious things. So we need to talk about CRISPR, not just as scientists or in academic journals or papers, but in public. And then, of course, because this, social media is democratic, people will talk about it in ways that are not true and scare people. And how do we fight that? Really complicated now with social media. So who are thought leaders that we can, someone like Fauci, we would say, is a scientific thought leader, and yet he's disparaged by the right. And, so how do we think about this? And at least we need to be talking about it. We just can't say, well, I'm going to accept whatever they do because they're scientists. They know what they're doing.
Yeah. Yeah.
I wish that were the case.
Well, I love how you define, I mean, you said science is a process. And I think that's the key, right? I mean, we like to think black and white, good and evil, but then there is many color, many shade of grays, and many things between the good and evil. And that's what ethics is all about. And as you started, we never talked about ethics, and philosophy has never been so popular since the classic Greeks, probably. Now with AI, the advanced technology and the power that we have into really, playing Mother Nature or God in changing the nature of who we are. So I think talking about it, it's extremely important. And I want to give you this last couple of minutes because on a topic like this, it's easy to just, time just fly having this conversation. Just a couple of points about your book. Who is it for? Who you had in mind when you wrote it? Is it for everyday people so they can start thinking about it? It's easy to digest, or more of an academic kind of book? And that's just a call to action to why you wrote this book.
Well, I wrote it. I'll start with your last question because
it's easier.
The first line in my book is CRISPR keeps me up at night, keeps me awake. Mm-hmm. Along with nuclear war, AI, climate change, things that an individual feels they don't really have control over. So how do we think about these things? And then from that point, I invited writers who are scientists, philosophers, bioethicists, parents, people, new voices, a trans bioethicist, Florence Ashley. No one's had asked a trans bioethicist to write about these things. An indigenous scientist, Crystal Tsosi, who is an indigenous woman from Arizona, who's a scientist, philosopher, and a cardiologist who has a child with albinism, a genetic mutation, someone with cystic fibrosis who faces the possibility of maybe getting cured, someone who's deaf, Carol Patton, who says, I don't see myself needing to be cured. Where do we draw the line on that? Is it just up to the parents to decide? Then we have a piece by someone who writes about animals and philosophically about animal rights and talks about transhumanism. You brought that up at the very beginning. We're implanting human genes, putting them into animals. Should we be making an animal more sentient? Is that possible? What does that mean? Or should we just make chickens that are have smaller and smaller heads and bigger bodies so that we make chicken, the meat? Where do we stand on how we deal with animals? It is enough to make you vegan when you read his piece about how animals are treated. And, we say, well, we want to make human lives better. But what are we doing to animals when we're making kidneys in pigs that can be transplanted because they have human elements? Can we start to transplant emotional things into, then they'll say that's science fiction, but, everything's been science fiction before, cars, planes, So our book is about opening up the discussion in many arenas, bioweapons, and then oversight, who's going to oversee this? Scientists have told me we don't need it because we'll monitor ourselves. We're good at that. That's questionable, arguable. And certainly with Dr. He, reading Ben's piece, you really start to wonder, because Ben argues that, or posits, that Dr. He did what he did because he was encouraged by some American scientists to go down that road. Dr. He was a fellow and a postdoc in the United States. And he gives a quote. I don't know what universities, I don't know the scientists. I don't think that's necessary for me to know, but he does quote a scientist. He was at a meeting with Dr. He and scientists, and they said, one person said, hire me for your company, but hire me as number 5, because it's going to be too controversial to bring me on as number 1 or number 2. Or they said to him, the person who started IVF, he didn't go down the traditional road of peer review. He announced it in the paper, and he won the Nobel Prize. So it's a complex situation of human aspiration, drive, fame-seeking, being proud, and promoting China, that many elements went into what Dr. He did that hadn't really been talked about before until our book, that Ben Hurlbut was privy to. So that's, I think, what's so interesting. It's divided, the book is divided into a number of different topics, like oversight and the future, new voices, unheard voices from the past, before, personal stories, new genetics, new eugenics, I should say. Are we about to just keep pushing and pushing to make people not have certain qualities? Who's deciding that? Michael Sandel, who's a professor at Harvard, wrote a piece years ago, about 20 years ago, in The Atlantic, where he talks about, his worry about there'll ultimately be the enhanced and the normal. And who's going to be enhanced? Are these going to be people who have money? We can't enhance people. As I said earlier, the genetics are way too complicated and too multifactorial. But that doesn't mean that we won't be able to do a lot of things in the future, like make people pain-free, or sleep less, or be, soldiers who have less fear, maybe.
There is a good sci-fi movie right there.
Well, it was called Gattaca. Many years ago, that, was very prescient about this.
Yeah, yeah. Wow, I think there is a lot to ponder, a lot to muse, a lot to think about, which it's my way of closing this episode, where I say, if you have more questions now in your head than at the beginning of this episode, I think we did a good job, because thinking is good. And it goes to show that even scientists, if they want to be objective, they realize that there is no final and perfect answers, because we are certainly, what make us human, I think, is just the fact that we're not perfect, which
Right.
will raise another question about, is this gen editing
Right.
Ruth aimed to make us something that we're not? But I'm not going to ask you this question because it's going to be another hour.
That's right.
I would love for you to come back
That'd be
again.
great.
Either talk about this here, because there is so much to talk about, but I'm more interested, well, in a different, and from a different angle, from a different perspective, I'm interested in the storytelling.
Great.
And,
We'll do
and that. So I'll have you back. For everybody listening, there will be link to the book, to Dr. Neal Baer website, and way to get in touch with him. I want to thank you very much.
Thank you.
For your time. It was an excellent conversation. My brain is going a million miles an hour, and I thank you for that.
Thank you for having me.
Thank you, everybody.
See you
Stay
soon.
tuned, subscribe, and I'll see you. And you will hear me on another story on the Redefining Society podcast. Take care.
You too. Bye.